anaerobic respiration, we are talking about this link reaction, right? Link reaction. So what is link reaction? Here the pyruvic acid, which is the 3 carbon compound, is getting converted to what is this? Acetyl CoA. This is a 2 carbon compound. So since it has to get balanced, there is one CO2 getting generated. Solradhu puriyudha? And the enzyme is going to be pyruvate dehydrogenase. As I said, wherever you see dehydrogenase, what is required? There is, see, dehydrogenase means there is it's a oxidoreductase. So what is oxidoreductase? There is going to be simultaneous oxidation and reduction. So this is going to lose the electron. Okay, so pyruvic acid is getting it loses the electron. So pyruvic acid oxidized. This is getting accepting electron, it becomes NADH. So this is going to get reduced. See, this also requires Mg2+. So without magnesium, this reaction cannot happen. So he said everything is important, right? And they have mentioned coenzyme A, NAD+. What are the other requirements for this reaction to happen? They have given, right? So we have done this link reaction. Now we will get into the tricarboxylic acid cycle, which is also otherwise referred as Krebs cycle. Krebs cycle, because the due discovery was done by a scientist called as Sir Hans Krebs, so it has got the name Krebs cycle, right? Okay, so again let us see here. It's a very interesting thing. So this is something different from your glycolysis, that this is a series of chemical reaction, but you know it starts with oxaloacetic acid and it ends with oxaloacetic acid. So this is a biochemical cycle. That's the reason why they have given it as citric acid cycle, right? So whereas glycolysis, your starting point is glucose and end point is pyruvic acid. So both are completely different. So it is called as a biochemical pathway. This is also a biochemical pathway, but it is linked. So it is called as a biochemical cycle, right? Okay. Now it has got 3 names. One is Krebs cycle. Okay, because due to the due credits to be given to the scientist who have given this cycle. So his name is Sir Hans Krebs. So Krebs cycle, and the first stable compound formed is citric acid. The first stable compound formed. There might be some intermediate compound formed in between, you know, this reaction to this reaction, right? But the first stable compound formed is a citric acid. So hence it is also called as a citric acid cycle, right? Okay, and most of the acids have got a 3-COOH group, tricarboxylic group, hence it is also called as tricarboxylic acid cycle. So it has got plenty of name and each and every name has got its own meaning. Purida, right? Gavani, pyruvate, right? Acetyl CoA, right? Acetyl CoA. Now this acetyl, you have to keep noting the number of carbon, right? One CO2 is lost here. This is a 2 carbon compound combining with oxaloacetic acid, which is a 4 carbon compound. So you get citric acid, which is a 4+2, which is a 6 carbon compound. As I said, you have got some intermediate reaction. Okay, so those are unstable. The first stable compound formed here is a citric acid, hence the name citric acid cycle, right? Now, clear arka? Now the enzyme is going to be citrate synthase. Right, citrate synthase, simple. Now listen, citric acid is becoming alpha keto glutaric acid, or it is also called as alpha keto glutarate. What is the number of carbon of citric acid? What is the number of carbon of alpha glutaric acid? 5. So 1 CO2 is lost here, right? And you also see the involvement of NAD+ to NADH. So what do you expect there to happen? Oxidation reduction. Reduction will happen. This can accept the electron only if someone is going to donate the electron. So this is something like your link reaction. Inga irukra reaction maari dhaan adu. Inga enna irukku par pyruvate reaction paathala. Inna mari puriyudha? Inga paru electron lose pannudu, CO2 lose pannudu. You are getting the point. Anamari, here you can see that this is citric acid is going to lose both electron, so it is getting oxidized. It is also leaving its CO2. What is the chemical term? Decarboxylation. So your citric acid is oxidatively decarboxylated to alpha ketoglutarate acid. and the electron is accepted by NAD plus and in turn it gets reduced to NADH. Clear இருக்கா? இப்போ ஒரு சின்ன question கேக்குறேன். From pyruvic acid, how many decarboxylation reaction you would expect from pyruvic acid? How many decarboxylation reaction you would expect from pyruvic acid? See, photosynthesis is carbon dioxide, okay, will get fixed as glucose. What is the formula of glucose? About how many carbon dioxide needed? 6. 6 CO2. 6 CO2. Now the glucose in respiration, the same glucose is getting liberated as carbon dioxide. So from one glucose, how many CO2 will get eliminated? Pyruvic acid has got how many carbon? Pyruvic acid is 1. What is it? 3 carbon. So how many CO2 should get eliminated? 3. 1 inga pedja, 1 inga pod. Now how CO2 pono? 1. Adhe mari there is another pyruvic acid because one glucose will give 2 molecules of pyruvic acid. Right? You are getting my point? But totally what is happening through this reaction, all the carbon in glucose is getting removed as carbon dioxide, whereas in photosynthesis all the carbon dioxide is getting fixed as glucose. சொல்றது புரியுதா? நான் என்ன சொல்றேன் கன்வே பண்றேன்னு புரியுதா? ரைட், ஓகே. இப்போ இங்க இது, ஸோ இங்க வந்து citric acid, alpha ketoglutaric acid. Now this alpha ketoglutaric acid will become, okay, you get an inga ஒரு product வரும், succinyl CoA ன்னு வரும் இந்த இடத்துல. You get a succinyl CoA, okay, where see here you get a product here, not here, inga இந்த இடத்துல வரும் succinyl CoA. Succinyl CoA, where again NAD plus is getting reduced to NADH and CO2. So what அப்போ succinyl CoA உடைய carbon எவ்வளவு இருக்கும்? What is the carbon of succinyl CoA? 4. Excellent. 4 carbon. Now this succinyl CoA becomes succinic acid or succinate. இங்க இருக்கும் அது புரியுதா? அவன் they have compressed and you have given the thing. So succinyl CoA will become succinic acid, 4 carbon. Here a GTP is formed. This is something similar to ATP, right? Something similar to ATP. Now succinic acid or succinate become malic acid. Nadula fumarate varum, again they have skipped that. So malic acid, where FAD, see here only you see an electron acceptor FAD plus other than NAD plus. Till now what you have seen, the one who is going to receive the electron is NAD plus, and here alone you see one exception where the electron acceptor is going to be FAD plus or not NAD plus. So FAD plus is getting reduced to FADH2, right? Succinic acid, then fumaric acid, malic acid, and malic acid becomes oxaloacetic acid. Again you see a presence of NAD plus getting reduced to NADH. So in short what is happening, you have a 4 carbon oxaloacetic acid, you get back the 4 carbon oxaloacetic acid. நான் சொல்றேன் 4 கார்பன் OAA இருக்கு, திரும்ப 4 கார்பன் OAA வந்துருச்சு. அப்போ what has happened to the acetyl CoA which has got added? நீ இது பார்த்தனா you just forget this. You start with OAA, this keeps on happening and you end with OAA. So 4 carbon is brought back. So what has actually, because here the acetyl CoA is what is going to combine with oxaloacetic acid, is going to give the citric acid. So what has happened to acetyl CoA? What has happened to acetyl CoA? நான் சொல்றது புரியுதா? See, totally you have got 6 carbon, 4 from here, 2 from here. This 4 you have got. What has happened to this 2? Removed as CO2. Again இல்லப்பா, removed as CO2. இந்த CO2 வெளில போகுதுல, அதெல்லாம் எங்க இருந்து வருது? புரியுதா? அப்போ pyruvic acid is leaving all its, see glucose, in short glucose is getting converted to 2 pyruvic acid. Pyruvic acid is leaving all its CO2. Totally a pyruvic acid of 3 carbon. 1 CO2 is last year, 1 CO2 is last year, 1 CO2 is last year. So in short, glucose to pyruvic acid is losing its carbon in the form of carbon dioxide. So all the carbon dioxide is getting liberated to the atmosphere. So this is what is respiration. Whereas in photosynthesis, you will see that all the CO2 will become glucose. CO2, in case if it become glucose, that is photosynthesis. CO2, if glucose is becoming CO2, that is respiration. How it becomes CO2? சொல்றது புரியுதா? Clear-ஆ இருக்கா? So now you can calculate ATP formed. Immediately mind அங்க போடும். How many ATP, how many SLPs are there? How many SLP in this Krebs cycle? SLP, you directly see the release of ATP or GTP. So how many? 1. How many oxidative phosphorylation? How many? How many NADH? 1, 2. NADH is 6. 1, 2, 3. So 3 into 1 NADH yields how many ATP? 3. So totally how many ATP you get? 3 into 3 is 9, dear. 3 into 3 is 9, right? So how many FADH do you have? Here, this is one FADH2. So 1 FADH2 yields 1 into 2 ATP. So it will be 2. So 9 plus 2 plus 1 is going to be how many ATP totally? We are going to have 12 ATP. So one Krebs cycle yields how many ATP? 12 ATP. Clear arka? Now, what is the case if number of ATP from one glucose molecule getting oxidized aerobically? How many ATP? Glycolysis, how many ATP? 8. Link reaction from one glucose. Link reaction, you have one NADH. Then which yields how many ATP? How many link reaction is happening? 2. So 6, 14. One Krebs cycle yields how many ATP? 12. 12. Memory 12. How many Krebs cycle?
1.
From one glucose, how many pyruvic acid?
2 pyruvic acid.
How many acetyl CoA?
2 acetyl.
So how many Krebs cycle?
2 Krebs cycle.
So 12. Each Krebs cycle yields how many ATP?
12.
அப்போ மொத்தமா எவ்வளவு வரும்? 12 into 2 is going to be 24. So totally how many, what is the yield here? 8 plus 6 is 14. 14 plus 24 is going to be 38. 38 ATP. Vrida? 38 ATP. So one molecule of glucose is going to yield you how many ATP? 38. Provided if it undergoes an aerobic respiration. Things are going to be completely different for anaerobic. சொல்றது புரியுதா? Actually, I have an argument to say it is 36, but your CBSE book says 38. So let's stop here. So 38. Clear arka? So one glucose molecule is going to yield you 38 ATP. புரியுதா? Clear arka? Right. Okay. Now we are going to talk about the electron transport chain, right? Electron transport chain. So listen dear, the electron transport chain is going to happen in mitochondrial membrane, whereas where has the Krebs cycle has taken place? Cytoplasm illappa, cytoplasm glycolysis எங்க நடக்குது? Krebs cycle எங்க நடந்திருக்கு? What, see, you have to be thorough with this once it has been thought. The confusion varthukku vaaipe kedaiyadu. It means that you have not given proper attention to it. Solradhu puriyudha? I am asking very basic things. See, glucose to pyruvic acid, where it has happened, cytoplasm, pyruvic acid is formed. In presence of oxygen, pyruvic acid will enter into mitochondria. So where do you expect Krebs cycle to happen? Mitochondria, இப்போ மைட்டோகாண்ட்ரியாக்குள்ள போனால் இப்போ நீ கிளாஸ்க்குள்ள இங்கே வந்தால் இங்கே தானப்பா கிளாஸ் இருக்கும். அந்த மாதிரி பைருவிக் ஆசிட் ஹஸ் காட் இன்டு மைட்டோகாண்ட்ரியா, வேர் டு எக்ஸ்பெக்ட் மைட்டோகாண்ட்ரியல் மேட்ரிக்ஸ். So where do you expect this to happen, electron transport chain to happen? Cristae. What is cristae? Inner foldings of mitochondrial membrane. So if I கொடுத்துட்டா this is the inner mitochondrial membrane, this is the matrix. So here where you expect a Krebs cycle to happen, புரியுதா? And this is the intermembrane space, this is the inner membrane, this is the outer membrane. So you have a space between here which is called as a intermembrane space. புரியுதா? intermembrane space, right? You are getting clear? இதெல்லாம் clear-ஆ இருக்கணும், right? Matrix mitochondriaல the central part is called as a matrix, whereas it has got a outer two membrane, right? Now this electron transport chain, it has got some complexes, like you call as a complex 1, you have a complex 2, you have, let me talk about that. Okay, here you have got a complex 2, then there is a complex 3, then there is a complex 4, right? Then there is also, okay, this is what is called as an oxysome. It has got a F0 particle, F1 particle. So let me talk about all those things. So in short, what is going to happen here is you have two electron ரெண்டு electron rich molecule இருக்கு, which is formed in the previously discussed metabolic reaction. So what are those two molecules? Electron rich molecule என்னது? என்கிட்ட ஒரு ரெண்டு இருக்கு, that is going to give electron to this molecule and get ATP. So what are those two molecules? எது இப்போ எலக்ட்ரானை பிடிச்ச குளுக்கோஸ்லேருந்து எலக்ட்ரானை வாங்கிட்டு இருக்கிறது மாலிக்யூல் யாரு? At various steps, some molecule have got the electron, obtained the electron from at various steps in the glucose metabolism, glucose oxidation. So what are those two molecules? NADH and FADH2. So both are going to give its respective electron to this molecule, in turn get ATP. So NADH, if it gives electron, is going to give 3 ATP. FADH is going to give electron, is going to give 2 ATP. Actually everything will be in the concept வந்து இங்க கரெக்ட்டா இருக்கும், return partல பக்சா கரெக்ட்டா இருக்கும், but here there is some confusion, right? So let's see, right? So in short, it's going to have complex 1, this is complex 2 here, this is complex 3, even I can put complex 2 here. Okay, ஏன் இப்படி கொடுத்திருக்காங்கன்னு தெரியல, right? So அதுக்கப்புறம் you have got a complex 4. So each complex has got a name, say NADH dehydrogenase, cytochrome BZ1, right? Succinate dehydrogenase, then cytochrome C oxidase. So each complex has got its own name. Clear இருக்கா? இப்போ you also see UQ molecule, தெரியுதா இங்க? UQ stands for ubiquinone. இதோ இருக்கு பாரு, ubiquinone. UQ na ubiquinone. So ubiquinone is the one which connects complex 1 and complex 3. புரியுதா? அதுக்கப்புறம் அதே தான் இங்கேயும் இருக்கு, அதே மாதிரி இங்க பாரு complex 3 யும் complex 4 யும் கனெக்ட் பண்றது cytochrome C. So ரெண்டு connecting molecule இருக்கு, one is going to be ubiquinone, other is going to be cytochrome C. புரியுதா? Right, listen ma, listen. So இதை ஞாபகம் வச்சுக்கோ, matrix, inner membrane, outer membrane, between inner membrane and outer membrane there is a space here which we call it as an intermembrane space. சொல்றது புரியுதா? இப்போ see NADH, it is giving the electron here, தெரியுதா இங்க electron. So NADH is oxidized to NAD+. சொல்றது புரியுதா? So what will be the fate of this NAD+? What is your guess? What is the fate of this NAD+? It gets released, it goes to Krebs cycle, get another electron. Make sense? Yeah. In the electron வந்து என்ன ஆகும்? The one which is released from NADH, so this electron comes from glucose. See, when you say pyruvic acid or when you say some step in pyruvic acid or some step in Krebs cycle, the electron actually comes from glucose. Glucose ல இருந்தா வருது, glucose to glucose 6-phosphate, a series of chemical reaction. So whatever, wherever step you see electron getting released, electron source on the glucose, and the electron இப்போ NADH வழியா வந்து it is entering into the complex. சொல்றது புரியுதா? கிளியரா இருக்கா? இப்போ இங்க என்ன ஆகுது, இந்த electron, let me make it very simple, electron will keep traveling full-ஆ போவோம். For the sake of better understanding, put the complex 2 here. So let us not disturb complex 2 now. As of now, with respect to NADH, we are not bothered about complex 2 now. So from electron from NADH will enter to the complex 1 and ubiquinone. ubiquinone, once it accepts the electron, ubiquinone will get reduced. உனக்கு ஏன் அந்த பிரச்சனை போலேருந்து இருக்குன்னு தெரியல. It gets reduced. Accept பண்ணா reduce ஆகும். So it becomes ubiquinol. இங்க கொடுத்துருக்கு பாரு, ubiquinol. Reduced ubiquinone is called as ubiquinol. புரியுதா நான் சொல்றது? எலக்ட்ரான் அப்புறம் என்ன ஆகும்? இங்க பாரு, when electron from ubiquinol goes to complex 3, this again gets oxidized to ubiquinol. புரியுதா நான் சொல்றது? கிளியரா இருக்கா? அதுக்கப்புறம் it comes to cytochrome 3. இதுல each and complex 3. Complex 3 ல you have to remember the electron carrier. அதாவது இது வந்து individual molecule. Here you have got some individual molecule which form a complex. அதனால தான் complex னு கொடுத்திருக்கான். அதுல cytochrome B இருக்கு, ferrous sulfur protein, cytochrome C1. ஸோ இந்த ஆர்டர்லாம் பார்த்துக்கணும். புரியுதா? அது வழியா வந்து அதுக்கப்புறம் எலக்ட்ரான் எங்க வருது சொல்லி இண்டிவிஜுவல் மாலிக்யூல் cytochrome C. Then cytochrome C லேருந்து electron goes to complex 4. You have got a cytochrome A. You have got, I'll make it a different color, right? You have cytochrome A. You have cytochrome A3 and finally copper, and இங்க வந்து actually oxygen வரும். இங்க இந்த இடத்துல oxygen வரும். Final electron acceptor is the oxygen there. சொல்றது புரியுதா? கிளியரா இருக்கா? I'll repeat it again. So NADH is going to give electron to complex 1. Complex 1 has got electron FMN, flavin mononucleotide. Flavin இங்க கொடுத்திருக்கா என்னன்னு தெரியல. flavin mononucleotide அப்படின்னு பேரு காம்ப்ளெக்ஸ். அந்த காம்ப்ளெக்ஸ் உள்ள FMN க்கு flavin mononucleotide, FLAVI, flavin riboflavin mononucleotide. FES na ferrous sulfur protein. அங்க இருந்து எங்க வரும்? individual molecule. What is that? Ubiquinone. Then to ubiquinone will become ubiquinol and accepting electron, right? Then complex 3. Complex 3 you have got cytochrome B, FES and cytochrome C1. அது mostly cytochrome ங்கறனால it's called a cytochrome BC1 complex. Cytochrome B here, cytochrome C1 here. From that to cytochrome C, right? Then it goes to complex 4 and finally there is an oxygen which is going to accept the electron. இந்த கொடுத்திருக்கு பாரு, oxygen plus proton, finally you give water. So water is the final product. So this இப்ப நம்ம inhale பண்ற oxygen எங்க சார் யூஸ் ஆகுது? நம்ம we have seen that case in breathing and exchange of gases. Oxygen இருக்கு, it is we inhale oxygen, it is getting exchanged in alveoli. From there it get transported via blood, then it goes to tissue oxygen from blood reaches the cells. Where is oxygen used in the cells அப்படின்னா இங்க தான். Last step of your internal respiration needs oxygen. இதுல இன்னொன்னு பார்த்தனா before the utilization of oxygen, CO2 is evolved. இதுக்கு முன்னாடியே whether the link reaction or in Krebs cycle which happens before this step, CO2 has already gone to the atmosphere, decarboxylation. So அதுக்கப்புறம் தான் உனக்கு இங்க O2ங்கிறதே யூஸ் ஆகுது. நான் சொல்றது புரியுதா? See, we always think whatever O2 actually before reading this chapter in my younger days I used to think whatever oxygen we consume is converted to oxygen. No, carbon dioxide. This is not the case actually. O2 has got a final role before which CO2 is evolved. Water la CO2 is evolved. Evolved na vella poidichu. Now you are getting my point what I am trying to right. So this is the point, right? Ipo gavan idu clear ah irukka? So you have to suppose to understand what are the complex involved. All the complex are involved except complex 2 with respect to electron from NADH. And you should also know what are the electron carrier molecule in each complex and what is the complex name. So everything is important, right? Now simple thing, electron transport across complex, leave about the individual molecule. Electron transport across complex is going to pump proton. Okay, proton. What is this region of mitochondria referred as? Matrix. This is the intermembrane space, this is the outer membrane, and the bluish one is the intermembrane. So whenever the electron passes, okay, across the complex, there is a proton getting from matrix to the intermembrane space. சொல்கிறது புரியுதா? proton வந்து automatically இப்போ இது வழியாக electron transport ஆச்சுன்னா proton from the matrix will get shuttled to the intermembrane space. அதே மாதிரி இங்கே பாரு, தெரியுதா? This doesn't happen when the electron passes to the individual molecule, only in the complex. அதே மாதிரி இங்கே பாரு, தெரியுதா? Right? அப்போ you see a particular number of protons, say 4 proton here. Oh, I'm sorry. You see a particular number of protons, say 4 proton here, 2 proton here, some proton. Actually, this has got some calculation. We are not bothered about that. Like how many protons should get pumped from matrix to the intermembrane space. But the point is, there is at our level there is some proton which is getting pumped from matrix to the intermembrane space. Clear irka? Appo this is going to form a gradient. Okay, this is going to form a gradient. Gradient na, you have a region, one side of the region there is going to be something in higher concentration, on other side of the region there is going to be something with a lower concentration. நான் சொல்றது புரியுதா? higher concentration, lower concentration. இதுக்கு பேர் தான் gradient ன்னு பேர். இப்போ இங்க என்ன gradient form ஆகுது? Gradient can be chemical. For example, I draw a line, I put some salt, there is no salt there, or 1 gram of salt, or 0.5 gram of salt, or 0.25 gram of salt. So there is a gradient. Either a decrease in concentration or increase in concentration. Gradient na adha. Ipo inga gradient form avudhu. Gradient with respect to which one? Proton. When all the protons are pumped, here actually there is a technically less proton. Since every proton is pumped here, in the intermembrane space will have a more proton. அதாவது inner membrane வழியா proton pump ஆகுது. Matrix of proton numbers, for example, there is going to be 10 proton அப்படின்னா, now 4 plus 4, 8, 8 plus 2, 10. Let's assume there is going to be 12 proton. Now only 2 is here, remaining 10 are in the intermembrane space. புரியுதா? Clear இருக்கா? Intermembrane space will see more number of proton compared to matrix. Is that clear? About what happens here? What happens? There is a proton gradient formed. Clear irka? There is a proton gradient formed, right? Appo there is going to be less proton here, there is going to be more proton there. Clear irka? Ipo at any point keep this as a rule. If there is going to be a proton, I mean if there is going to be a break in the gradient உடைஞ்சதுன்னா energy will be released. அப்போ இங்க say 10 proton இருக்குன்னு வெச்சுப்போம், right? Now this F0 particle னு இருக்குல்ல இங்க, this is what is the proton pump. Now when this proton pump, through this proton pump, if the 10 proton gets to the matrix, சொல்றது புரியுதா? அப்போ there is a break in proton gradient. புரியுதா? you have some barrier, இங்க தண்ணி நிறைய இருக்கு, இங்க தண்ணி கம்மியா இருக்கு. அப்போ I said there is a water gradient. If I am going to make a hole, now water from this part higher concentration, அப்போ I am breaking the water gradient. That is what people use it in dam and make it as an hydroelectric power. Energy release ஆகும். புரியுதா? I am not sure about the physics and chemistry part of it, but energy release ஆகும். Clear இருக்கா? அந்த மாதிரி what happens is your this is the F0 is the proton pump. So whatever proton that is accumulated in the intermembrane space will get it back to the matrix. அப்போ energy release ஆகுது. அந்த எனர்ஜியை இந்த F1 particle இருக்கு தெரியுமா? F1 particle has got one enzyme activity which is called as ATP synthase. கொடுத்துருக்கு, அங்கேயே கொடுத்துருக்கு ATP synthase. அப்போ அது என்ன பண்ணும்? whatever energy which is released due to the the break of the proton gradient will be captured by the ATP synthase and make it as ATP. இதோ கொடுத்துருக்கு பாரு, ADP plus PI will give you ATP. What sort of chemical reaction is this? Phosphorylation. Because PI na inorganic phosphate. What does PI stands for? Inorganic phosphate. So this inorganic phosphate combines with ADP and form ATP. இங்க பாரு, ஒரு நிமிஷம், இதுக்கு எனர்ஜி தேவை. Okay, இதுக்கு எனர்ஜி தேவை. The energy ATP synthase இருந்ததுன்னா it can do this. That energy is obtained by the release of the proton. And that release of proton is actually done by the proton pump, which is nothing but F0 particle. Why there is a release of energy? Because there is a break in proton gradient. Why there is a break in proton gradient? It is due to the passing of proton from matrix to intermembrane space due to transfer of electron from NADH. இது clear-ஆ இருக்கா? இப்போ நான் என்ன பண்ணுறேன், I'll also do the FADH2 part. Then we will have a doubt clarification or discussion வச்சுக்கலாம், right? இப்போ you put complex 2 technically should be above complex 3. I don't know why they have given below it. புரியுதா? complex 2ங்கிறது இங்க என்ன கொடுத்திருக்கான் பாரு, succinate dehydrogenase complex. Actually this is a part of Krebs cycle. நல்லா கவனி, Krebs cycle இது உண்டு, it's a part of Krebs cycle, right? இப்போ what happens, இது வந்து இந்த complex 2ங்கிறது is not like complex 3 or complex 4 மாதிரி கிடையாது. அது ஒரு chemical reaction, that is இங்கே கொடுத்திருக்கான் பாரு, complex 2 தானே, succinate to fumarate. இதுதான் நடக்குது Krebs cycle. புரியுதா? So it is the complex 2 which is releasing FADH2. நான் சொல்றது புரியுதா? In short ஞாபகம் வச்சுக்கோ, this complex 2, right, complex 2 this complex 2 where FADH2 is produced gives the electron to FES. What is FES there? Ferrous sulfur protein. அந்த electron அப்போ இது வந்து this become FAD plus ஆயிடும். புரியுதா நான் சொல்றது புரியுதா? So electron from FES எங்க போகுது? ubiquinol அங்க போயிட்டு finally complex 3க்கு போயிடுது இது வழியா. அதாவது நான் சொல்றேன் கேளு, இந்த complex 2 இங்க போட்டாலும் இங்க தான் ubiquinolல ஜாயின் ஆகும். இந்த complex 2 இங்க போடுறேன்னா எலக்ட்ரான் வந்து ubiquinolல போகும். புரியுதா நான் சொல்றது? இங்க போட்டாலும் ubiquinolல தான் போகுது. It looks that from complex 2, okay, electron should go to ubiquinol. அவன் இங்க ரெண்டு ubiquinol வச்சிருக்கான், இங்கே அதே தான் வச்சிருக்கான். புரியுதா? ubiquinol வந்து complex 3க்கு வரும். சொல்றது புரியுதா? Now what happens, ubiquinol இங்க இருந்து complex 3, so ஒரே pathway complex 3, then you have எங்க போகுது cytochrome C, இதான் pathway. இதுலேருந்து இப்படி pathway, இதுலேருந்து இப்படி போயிட்டு it is come to cytochrome C, then it is come to complex 4, then அவ்வளவுதான் water. புரியுதா? அப்போ இங்க active complex ன்னு பார்த்தா complex 3 and complex 4 which is ஏன்னா FADH2 ல இருந்து complex 1 இன்வால்வ் ஆகாது. நீ கவனிச்சு பாரு, FADH2 ல இருந்து you see this is complex 2, complex 3 இன்வால்வ் ஆகுது, okay, then complex 4 இன்வால்வ் ஆகுது, complex 1 இன்வால்வ் ஆகாது. NADH ல இருந்து complex 2 இன்வால்வ் ஆகாது வரக்கூடிய எலக்ட்ரான். சொல்றது புரியுதா? you have to know how from complex 2 ubiquinol, complex 3, complex 4, from NADH complex 1 ubiquinol, complex 3, complex 4. அந்த சீக்வென்ஸ் தெரிஞ்சுக்கணும். புரியுதா? ஓகே, இதுதான். இது இங்க போடலாம் அந்த சீக்வென்ஸ் சேம் தான். புரியுதா? நான் ஒருவேளை திரும்ப சொல்றேன், complex 2 இங்க போட்டிருந்தா டைரக்டா ubiquinolல கொடுத்திருப்பேன். அவங்க இன்னொரு ubiquinol கொடுத்து இங்க கொடுத்திருக்கான். You are getting my the point I tried to convey. அப்ப இங்க என்ன ஆகும்? proton eliminate ஆகும். I mean intermembrane space க்கு shuttle ஆகும். So proton gradient is formed. So that proton gradient, okay, once it get released due to the proton exit into the matrix through the F0 particle, okay, there is a proton gradient break releasing energy. That energy is used by the ATP synthase, which is used in making the ADP plus PA giving rise to ATP. இதுல என்ன ஒண்ணுனா, one NADH molecule gives how many ATP? 3. One FADH2 molecule give how many ATP? 2. அந்த டிஃபரன்ஸ் ஏன் அப்படின்னா, ஓகே, இதுல பார்த்தனா உனக்கு காம்ப்ளெக்ஸ் 1ல வர புரோட்டான் வந்து வராது FADH2 ல இருந்து, because காம்ப்ளெக்ஸ் 1 அவாய்ட் பண்ணுது. அப்போ only actively complex 3 and complex 4 is involved. அப்போ less number of proton getting shuttled to the intermembrane space. Whereas if when you consider electron from NADH, you have this yellow, you have this green, and you have this complex 3, I mean complex 4, which is the red. அப்போ more இப்போ 10 புரோட்டான் வரும் பா, 4 according to this diagram, 4 4 8 8 2 10. எப்போ electron from NADH. FADH ல பார்த்தனா complex 2 doesn't give any proton. complex 3 ல 4, complex இதுல வந்து what is that 4 ல 2. so 4 proton less in intermembrane space compared to matrix. நான் சொல்றது புரியுதா? ஷார்ட்டாக சொன்னோன்னா NADH electron transfer will put 10 proton from matrix to membrane space because it involves actively complex 1, complex 3 and complex 4. Whereas FADH2 involves actively only complex 3 and 4. How about complex 2? Complex 2 is just a reaction of Krebs cycle. It is not pure complex, right? Appo it is going to put only 6 electron from matrix 2. Appo less number of proton with respect to FADH2 that is getting transferred to the through the pump. Appo obviously 2 ATP, whereas your NADH will eat 3 ATP. இதுதான் கான்செப்ட். புரியுதா? கிளியரா இருக்கா? இது ஆக்சுவலாக 2-3ங்கிறது இப்போ நம்ம படிக்கிறோம். But as I said yesterday, previous class, once you go to higher standards, you will be seeing that என்ன சொல்கிறது 2.5 ATP, 1.5 ATP higher level thinking மாறும். But here we are supposed to understand what is there in the NCERT text. கிளியரா இருக்கா?